Assalamualaikum. Hi guys. We continue our lesson about glucan, still in chapter number 3. Okay, as you have learned before, okay, so glucan is actually uh, amorphous, okay, metric components. Is a polymers of glucose, and you also have learned about this structure, the location, and of course this is different types of fungus. Glucan is here, and down there glucan is here, the left hand side. Okay, so again, glucans are the yellow color, okay, above of this structure of the cell wall component. Okay, so again, glucan, most fungal walls contain beta change glucan. Glucan are polysaccharides, okay, of course they are carbohydrate that contain only glucose as structural components and link with beta glycosidic bonds. Walls of ascomycete and basidiomycetes contain beta-1,3 glucan with branching of beta-1,6 glucan. There are some fungi with alpha-1,4 glucan. So different type of fungus, they have their own one. So if you look at this picture, you know, your right hand side here, this is the shiitake mushroom, contains of beta glucan. So that's why a lot of Japanese, you know, and Chinese, you know, they like it because they have their own benefit to them. Okay, so again, this is a structure of beta glucans. Again, beta glucans are polysaccharides of D-glucose monomers linked by beta glycosidic bonds. Okay, this is a diagram showing orientation and location of different beta glucan linkage. Okay, glycoprotein and proteins. Glycoproteins are proteins that contain oligosaccharides chain or glycans covalently attached to the polypeptides side chains. Glycoproteins include monoproteins. Galactoprotein and xyloproteins are important components of the matrix of the cell walls. Glycoprotein contains of glycosamine and N-acetylglucosamine. In parasitic fungi such as Candida albicans and Aspergillus fumigatus, the glycoproteins are antigens. Monoproteins in Saccharomyces cerevisiae are large molecules. Okay, again about glycoprotein and protein. Hydrophobin is a very hydrophobic proteins and found in area hypae of schizophyllum commune and are components of rodlets. Rodlets are found on the surface of conidia of Neurospora, Casa, and Aspergillus nidulans and protects the hypae from desiccation. Okay, constructions. Wall constructions take place in the apical tips. Autoradiographic studies suggest that all synthesis of chitin and glucans take place within 1 mm of the apex. The tip is highly plastic as the wall is laid down, walls rigidly with maturity. The rigidity is provided by cross-linking of polymers, thickening of fibrils, and deposition of material in the interfibrillar matrix. The process is highly polarized and reliant on maintenance of a positive Turgor pressure within the cytoplasm. So the wall have a number of characteristics which can be attributed to components of the wall. For instance, hypae are generally resistant to loss of water and yet capable of detection of signal for mating and conjugation between compatible hypae. The function of hydrophobins is typically of dysfunctional complexity located in the wall. Okay, next function is a communication. Okay, conjugation such as in anastomosis and mating reaction in fungi are similar, different but compatible glycoproteins called agglutinins in the wall of rich of each complementary hyphae fuse to form a complex binding the cells together. In mating, a release of hormone-like compounds proceeds the bindings. The hormone alters receptor glycoprotein in the wall, leading to a cascade of changes to the fibrillar construction of the cell wall. Ultimately, the wall breaks down. A conjugation tube may connect the walls. Membranes fuse, linking the cytoplasm of the two cells. So this is actually about communication. Okay, next one is continue about communication as well. Adjacent is also mediated by fibrillar glycoprotein subbundle of a gel-like matrix. Fibrils are commonly found where fungi attach to surfaces. The fibrils are highly specific in where they attach, and a complementary protein on the surface of the partner is assumed. This system of recognition and communication between fungi and partner is widespread in pathogenic and mutualistic interaction. This is actually for those particular fungus that are very dangerous one or pathogenic one. Okay. This is about hydrophobins in wall. They are large, diverse group related proteins found widely and only in the fungi. They may constitute up to 10% of total wall protein. Each molecule consists of hydrophobic domain and hydrophilic domain. They like water and they don't like water. Okay, the characteristic. The amphithetic structures provide the molecule with extraordinary potential array for function for the fungus, and in biotechnology, the presence of hydrophobins may also increase the strength of the cell wall. So I think we have learned about this one before. You have learned about this one from the previous lecture or lesson. Okay. So as a conclusion, fungal walls consist of complex fibrillar material abundant in polysaccharides and other components, compounds and functional complexity associated proteins and glycoproteins. The wall may be highly protected or relatively susceptible to the environment because of various constituents in the matrix. The wall also allows the cell to communicate with the environment, enabling reproduction, recognition, and reception. So this is the conclusion about cell wall. So for revision, as you can see from here, there are alphabet A to F, a structure of the fungus and their own characteristic. I think you may take a look later on. Okay, but please learn about this one. A is actually blastoconidia formation, B fission formation, C pseudomyphidio-hyphae, D connective hyphae, E septate hyphae, and the last one F septate hyphae with a clamp connection. So as a revision, this is actually a comparison between type of structure of hyphae. Okay, so please take a look and please make sure you know how to differentiate. Okay, I think that's all. Thank you very much.